Hi class, this is your instructor, Skylar Huff. We're now in Chapter 14, DNA, the genetic material. The rediscovery of Mendel at the turn of the 20th century led to the discovery of the genetic mechanisms detailed in Chapters 12 and 13. One question not directly answered for more than 50 years was perhaps the simplest. What are genes made of? Genes were known to be on chromosomes, but they are complex structures composed of DNA, RNA, and protein. This chapter describes the chain of experiments that led to the model for DNA pictured here before you, and along with it, the molecular mechanisms of heredity. This model was proposed by James Watson and Francis Crick, but was based on data from many others. These experiments are among the most elegant in science and represent the dawn of a molecular era still with us today. Back in chapters 12 and 13, you learned about the nature of inheritance and how genes, which contain the information to specify traits, are located on chromosomes. This finding led to the question of what components of chromosomes actually contains genetic information. Specifically, biologists wondered which molecule carries genetic information. They knew that chromosomes are comprised primarily of both protein and DNA. Which of these organic molecules actually makes up the gene? Starting in the late 1920s and continuing for about 30 years, a series of investigations addressed this question. DNA consists of 4 chemically similar nucleotides. In contrast, protein contains 20 different amino acids that are much more chemically diverse than nucleotides. These characteristics seemed initially to indicate greater informational capacity in protein than in DNA. However, experiments began to reveal evidence in favor of DNA. We describe 3 of those major findings now in this section. The first clue came in 1928 with the work of British microbiologist Frederick Griffith. Griffith was trying to make a vaccine that would protect against influenza, which was thought at the time to be caused by the bacteria Streptococcus pneumoniae. There are 2 forms of this bacteria: the normal virulent form that causes pneumonia and a mutant non-virulent form that does not. The normal virulent form of this bacterium is referred to as the S strand or S form because it forms smooth colonies on a culture disc. The mutant non-virulent form, which lacks an enzyme needed to manufacture the polysaccharide coat, is called the R form or rough form because it forms rough colonies. Griffith performed a series of simple experiments in which mice were infected with these bacteria, then monitored for disease symptoms. Mice infected with the virulent S form died from pneumonia, whereas infection with the non-virulent R form had no effect. This result shows that the polysaccharide coat is necessary for virulence. If the virulent S form is first killed with heat, infection does not harm the mice, showing that the coat itself is not sufficient to cause disease. Lastly, infecting mice with a mixture of heat-killed S form with live R form caused pneumonia and death in the mice. This was unexpected, as neither treatment alone caused disease. Furthermore, high levels of live S form bacteria were found in the lungs of dead mice. Somehow, the information specifying the polysaccharide coat had passed from the dead virulent S bacteria to live coatless R bacteria in the mixture, permanently changing coatless R bacteria to the virulent S variety. Griffith called this transfer of virulence from one cell to another transformation. Our modern interpretation is that the genetic material was actually transferred between cells. The agent responsible for transforming Streptococcus went undiscovered until 1944. In a classic series of experiments, Oswald Avery and his co-workers, Colin MacLeod and Macklin McCarty, identified the substance responsible for transformation in Griffith's experiment. They prepared the mixture of dead S. Streptococcus and live R. Streptococcus that Griffith had used. Then they removed as much of the protein as they could from their preparation, eventually achieving 99.98 purity. And that's 99.98% purity. They found that despite the removal of nearly all protein, the transforming activity was not reduced. Moreover, the properties of this substance resembled those of DNA in several ways. 1. The elemental composition agreed closely with that of DNA. 2. When spun at high speeds in an ultracentrifuge, it migrated to the same level or density as DNA. 3. Extracting lipids and proteins did not reduce transforming activity. 4. Protein digesting enzymes did not affect transforming activity, nor did RNA digesting enzymes. 5. DNA digesting enzymes destroyed all transforming activity. So these experiments supported the identity of DNA as the substance transferred between cells by transformation and indicated that the genetic material, at least in this bacterial species, is DNA. Avery's results were not widely accepted at first because many biologists continued to believe that proteins were the genetic material. But additional evidence supporting Avery's conclusion was provided in 1952 by Alfred Hershey and Martha Chase, who experimented with viruses that infect bacteria. These viruses are called bacteriophages, or more simply, phages. Viruses will be described in more detail later in this course in chapter 26. And they are more simpler than cells. They generally consist of genetic material, DNA or RNA, surrounded by a protein coat. When a phage infects a bacterial cell, it first binds to the cell's outer surface and then injects its genetic material into the cell. Once inside the cell, the viral genetic information uses the cell's information processing machinery to make viral proteins, leading to the production of thousands of new viruses. The buildup of viruses eventually causes the cell to burst or lyse, releasing progeny phage. The phage used by Hershey and Chase contains only DNA and protein, providing the simplest possible system to differentiate the roles of DNA and protein. If they could identify the molecule injected into the cell, they would identify the virus's genetic material. To do this, they needed a method to introduce unique labels to both DNA and protein. Nucleotides contain phosphorus, but proteins do not. And some amino acids contain sulfur, but DNA does not. Thus, the radioactive 32P isotope will label DNA specifically, and the isotope 35S will label proteins specifically. The two isotopes are easily distinguished based on the particles they emit when they decay. 2 experiments were performed. In one, viruses were grown on a medium containing 32P, which was incorporated into DNA. In the other, viruses were grown on a medium containing 35S, which was incorporated into protein coats. Each culture of labeled viruses was then used to infect separate bacterial cultures. After infection, the bacterial cell suspension was agitated in a blender to remove the infecting viral particles from the surfaces of the bacteria. This step ensured that only the part of the virus that had been injected into the bacterial cells, that is, the genetic material, would be detected. Each bacterial suspension was then centrifuged to produce a pellet of cells for analysis. In the 32P experiment, a large amount of radioactive phosphorus was found in the cell pellet, but in the 35S experiment, very little radioactive sulfur was found in the pellet. Hershey and Chase deduced that the genetic information viruses injected into bacteria was DNA and not protein. This shows what they found. DNA was discovered in 1869 by Friedrich Miescher. This was only 4 years after Mendel's work was published and 30 years before Mendel was rediscovered. Miescher extracted a white substance from the nuclei of white blood cells. He also isolated the same substance from salmon sperm. Analysis of this material, which was rich in phosphorus, indicated that it appeared to differ from other known cellular constituents. Mesher named this new biological substance nuclein because it was associated with the nucleus. Later, because nuclein was slightly acidic, it came to be called nucleic acid. Although the three-dimensional structure of the DNA molecule was a mystery, the components of nucleic acids were known. 1. A 5-carbon sugar. 2. A phosphate group or phosphate backbone. 3. A nitrogen-containing base or nitrogenous base. This may be a 2-ringed purine, those are adenine or guanine, or a single-ringed pyrimidine, those are thymine or cytosine, and in RNA, the pyrimidine uracil replaces thymine. We need a way to be able to unambiguously refer to each carbon atom in a nucleotide. We do this by adding a prime symbol to carbon numbers in the sugar to distinguish these from atoms in the bases that are just numbered. The ribose sugars found in nucleic acids consist of a 5-membered ring with 4 carbon atoms and an oxygen atom. As you see before yourselves, the carbon atoms are numbered 1' to 5' clockwise from the oxygen atom. And please look closely at this figure. Oxygen is there at the top of that pentose sugar. So going clockwise, you have 1', 2', 3', 4', 5'. The phosphate group is attached to the 5' carbon atom of the sugar, and the bases are attached to the 1' carbon. In addition, a free hydroxyl group is attached to the 3' carbon atom. This means each nucleotide has a 5' phosphate end and a 3' hydroxyl end. Nucleotide monomers are joined together by a dehydration reaction involving the 5' phosphate of one nucleotide with the 3' hydroxyl of another nucleotide. This linkage is called a phosphodiester bond because the phosphate group is now linked to the 2 sugars by means of a pair of ester bonds. Literally hundreds of millions of nucleotides are joined together via these linkages to form the nucleic acid polymers in your DNA. And there you see that phosphodiester bond. Polynucleotide strands of DNA or RNA can be thought of as starting with a 5' phosphate group and ending with a 3' hydroxyl group. Therefore, every DNA and RNA molecule has an intrinsic polarity and can refer unambiguously to each end of the molecule. By convention, the sequence of bases is usually written in the 5' to 3' direction. I repeat, in the 5' to 3' direction. To understand the model that Watson and Crick proposed, we need to review evidence that they had available to construct their model. A careful study carried out by Urban Shargoff showed that the nucleotide composition of DNA molecules varied in complex ways depending on the source of the DNA. This strongly suggested that DNA was not simply a simple repeating polymer, and that it might have the information encoding properties genetic material requires. Despite DNA's complexity, however, Shargoff observed an important underlying regularity in the ratios of the bases found in native DNA. The amount of adenine present in DNA always equals the amount of thymine, and the amount of guanine always equals the amount of cytosine. 2 important findings from this work are often called Shargoff's rules. 1. The proportion of A always equals that of T, and the proportion of G always equals that of C, or A equals T and G equals C. And secondly, the ratio of guanine to cytosine to adenine to thymine varies with different species. As mounting evidence indicated that DNA stored the hereditary information, investigators began to puzzle over how such a seemingly simple molecule could carry out such a complex coding function. The technique of X-ray diffraction provided more direct information about the possible structure of DNA. In X-ray diffraction, crystals of a molecule are bombarded with a beam of X-rays. The rays are bent or diffracted by the molecules they encounter, and the diffraction pattern is recorded on photographic film. The patterns resemble the ripples created by tossing a rock into a smooth lake. When analyzed mathematically, the diffraction pattern can yield information about the three-dimensional structure of a molecule. The problem with using this technique with DNA was that in the 1950s it was impossible to obtain true crystals of natural DNA. However, British researcher Maurice Wilkins learned how to prepare uniformly oriented DNA fibers, and with graduate student Ray Gosling succeeded in obtaining the first crude diffraction information on natural DNA in 1950. Their X-ray photos suggest that the DNA model has the shape of a helix. The British chemist Rosalind Franklin continued this work, perfecting the technique to obtain even ever clearer pictures of this work, of the oriented DNA fibers, So what can I say? The British chemist Rosalind Franklin continued this work, perfecting the technique to obtain ever clearer pictures of the oriented DNA fibers. The clearest of these images, shown to you here, confirmed that DNA was a helix and allowed calculation of the dimensions of the molecule, indicating a diameter of about 2 nanometers and a complete helical turn every 3.4 nanometers. Topic: forms of bases. One piece of evidence important to Watson and Crick was the form of the bases themselves. Because of the alternating double and single bonds in the bases, they actually exist in equilibrium between two forms when in solution. The different forms have to do with keto versus enol groups and amino versus imino groups that are attached to the bases. These structural forms are called tautomers. The importance of this distinction is that the two forms have very different hydrogen bonding possibilities. The predominant forms of the base contain the ketoamine groups, but a prominent biochemistry text of the time actually contained the opposite and incorrect information. Legend has it that Watson learned the correct forms while having lunch with a biochemist friend. Learning informally of Franklin's results before they were published in 1953, American chemist James Watson and English molecular biologist Francis Crick, two young investigators at Cambridge University, quickly worked out a likely structure for the DNA molecule, which we now know was substantially correct. Watson and Crick did not perform a single experiment themselves related to DNA structure. Rather, they built the detailed molecular models based on the information available. The key to the model was their understanding that each DNA molecule is made up of two chains of nucleotides that are intertwined, the double helix, phosphodiester backbone. The two strands of the double helix are made up of long polymers of nucleotides, and as described earlier in this section, each strand is made up of repeating sugar and phosphate units joined by phosphodiester bonds. We call this the phosphodiester backbone of the molecule. The two strands of the backbone are then wrapped about a common axis, forming a double helix. The helix is often compared to a spiral staircase in which two strands of the double helix are the handrails on the staircase. Watson and Crick proposed that the strands were held together by hydrogen bonds between the bases on opposite strands. The nature of these hydrogen bonds results in specific base pairs. Adenine can form two hydrogen bonds with thymine in an AT base pair. Guanine can form three hydrogen bonds with cytosine in a GC base pair. That is shown to you here. Note that each base pair combines a two-ringed purine with a single-ringed pyrimidine, which means that the diameter of each base pair is the same. A purine-purine base pair would be wider, and a pyrimidine-pyrimidine base pair would be narrower. In either case, this would distort the double helix formed by base pairs. We call this pattern of base pairing complementary, and this is a critical feature of all aspects of DNA function. The two strands of the DNA molecule are not identical, but each strand specifies the other because of the complementarity of base pairs. This means that if you know one strand of the DNA double helix, you know the other strand, even though they are not the same. For instance, the DNA strand 5' A, G, G, C, T, A, T, G, C, T, A, G, C, T, C, C, T, T, A, 3'. You should write this down. We'll have the complementary sequence 3', T, C, C, G, A, T, G, A, T, T, C, G, A, G, G, A, T, T, 5'. If there was no mistake made, please check your text. I don't think I made a mistake. Nonetheless, we will see this principle of complementarity in action as we learn about DNA, replication, and expression in section 14.3. Coming up next. The Watson-Crick model also explains Chargaff's results. The reason that the amount of A equals T and the amount of G equals C is that A base pairs with T and G base pairs with C. The relative amounts of A, T, and G, C can vary, but the amount of A must be equal to T and the amount of G must be equal to C because of complementary base pairing. As stated earlier in this section, a phosphodiester strand has an inherent polarity, meaning that one end terminates in a 3' OH and the other end terminates in a 5' phosphate group. Strands are thus referred to as having either a 5 to 3 or 5' to 3' or a 3' to 5' polarity, or 3 to 5. Two strands would be put together in two ways, with the polarity the same in each, that's parallel, or with the polarity opposite, that's anti-parallel. Native double-stranded DNA always has the anti-parallel configuration, with one strand running 5' to 3' and the other running 3' to 5'. In addition to its complementarity, this anti-parallel nature also has important implications for DNA replication. In the Watson and Crick model, a DNA molecule consists of two phosphodiester strands wrapped about a common helical axis with the bases extending to the interior of the helix. The sequence of bases on the two strands is complementary. So they used to form base pairs that hold the strands together. Each strand is also polar, with a 5' and 3' end, and they are oriented in opposite anti-parallel direction. Although each individual hydrogen bond is low energy, the sum of thousands or millions of such bonds makes a very stable molecule. This also means that a DNA molecule can have regions where the helix can be opened up without affecting the stability of the entire molecule, which is crucial for its function. Although the Watson and Crick model provided a rational structure for DNA, researchers had to answer further questions about how DNA could be replicated, a crucial step in cell division, and also about how cells could repair damaged or otherwise altered DNA. The accurate replication of DNA prior to cell division is a basic and crucial function. Research has revealed that this complex process requires the participation of a large number of cellular proteins. Before, geneticists could look for these details. However, they needed to perform some groundwork on the general mechanism. The Watson-Crick model immediately suggested that the basis for copying the genetic information is complementarity. One chain of the DNA molecule may have any conceivable base sequence, but this sequence completely determines the sequence of its partner in the duplex. In replication, the sequence of parental strands must be duplicated in daughter strands. That is, one parental helix with 2 strands must yield 2 daughter helices with 4 strands. The 2 daughter molecules are then separated during the course of cell division. Three models of DNA replication are possible. In a conservative model, both strands of the parental duplex would remain intact, conserved, and new DNA copies would consist of all new molecules. Both daughter strands would contain all new molecules. In a semi-conservative model, one strand of the parental duplex would remain intact in each daughter duplex, semi-conserved. A new complementary strand would be synthesized with each parental strand. Each daughter duplex would consist of one parental strand and one newly synthesized strand. And lastly, number 3, in a dispersive model, copies of DNA will consist of mixtures of parental and newly synthesized strands. That is, the new DNA will be dispersed throughout each strand of both daughter molecules after replication. That is shown to you here. Number 1 is conservative, the conservative model. Number 2 is the semi-conservative model. And then number 3 is the dispersive model, as I just described briefly. Notice that these 3 models suggest general mechanisms of replication without specifying any molecular details of the process. The 3 models of DNA replication were evaluated in 1958 by Matthew Meselson and Franklin Stahl. To distinguish between these models, a labeled DNA then followed the labeled DNA through 2 rounds of replication. The label Meselson and Stahl used was a heavy isotope of nitrogen, 15N, not a radioactive label. Molecules containing 15N have a greater density than those containing the common 14N isotope. Ultracentrifugation can be used to separate models that have different densities. Bacteria were grown in a medium containing 15N, which became incorporated into the basis of the bacterial DNA. After several generations, the DNA of the bacteria was denser than that of bacteria grown in a medium containing normally available 14N. Meselson and Stahl then transferred the bacteria from the 15N medium to the 14N medium and collected the DNA at various time intervals. The DNA for each interval was dissolved in a solution containing a heavy salt, cesium chloride. This solution was spun at very high speeds in an ultracentrifuge. The enormous centrifugal forces caused cesium ions to migrate toward the bottom of the centrifuge tube, creating a gradient of cesium concentration, and thus of density. Each DNA strand floated or sank in the medium until it reached the point at which its density exactly matched the density of cesium at that location. Because 15N strands are denser than 14N strands, they migrated farther down the tube. The DNA collected immediately after the transfer of bacteria to new 14N medium was all of one density equal to that of 15N DNA alone. However, after the bacteria completed the first round of DNA replication, the density of their DNA had decreased to a value intermediate between 14N DNA alone and 15N DNA. After the second round of replication, 2 density classes of DNA were observed, one intermediate one and one equal to that of the 14N DNA. Meselson and Stahl compared their experimental data with the results that would be predicted on the basis of 3 models. The conservative model was not consistent with the data because after one round of replication, 2 densities should have been observed. DNA strands would either be all heavy, parental, or all light, daughter. This model is rejected. The semi-conservative model is consistent with all observations. After one round of replication, a single density would be predicted because all DNA molecules would have a light strand and a heavy strand. After 2 rounds of replication, half of the molecules would have 2 light strands, and after 2 rounds of replication, half the molecules would have 2 light strands, and half would have a light strand and a heavy strand. So 2 densities would be observed. Third, the dispersive model was consistent with the data from the first round of replication because in this model, every DNA helix would consist of strands that are mixtures of half light, which is new, half heavy, which is old molecules. But after 2 rounds of replication, the dispersive model would yield only a single density. DNA strands would be composed of 3/4 light and 1/4 heavy molecules instead of 2 densities. Instead, 2 densities were observed, therefore this model is also rejected. The basic mechanism of DNA replication is semi-conservative. At the simplest level, then, DNA is replicated by opening up a DNA helix and making copies of both strands to produce 2 daughter helices, each consisting of 1 old strand and 1 new strand. And this is showing you the rounds of Mendelssohn-Stahl's experiment. And now let's get to the process of replication. Replication requires 3 things: something to copy, something to do the copying, and the building blocks to make the copy. The parental DNA molecules serve as a template. Enzymes perform the actions of copying the template, and the building blocks are nucleotides. Note that nucleotides have 1, 2, or 3 phosphate groups, which are called nucleoside mono, di, and triphosphates. The nucleoside triphosphates are the actual building blocks, and during synthesis, the 2 terminal phosphates are cleaved, providing energy for the reaction. DNA replication has a beginning, where the process starts, a middle, where the building blocks are added, and an end, where the process is finished. For biochemical processes like replication, we call this initiation, elongation, and termination. Although these divisions may seem arbitrary, in fact, discrete functions are usually required for initiation and termination that are not necessary for elongation. A number of enzymes work together to accomplish the task of assembling a new strand, but the enzyme that actually matches existing DNA bases with complementary nucleotides and then links the nucleotides together to make the new strand is DNA polymerase. All DNA polymerases that have been examined have several common features. They all add new bases to the 3' end of the existing strands. That is, they synthesize in a 5' to 3' direction by extending a strand of base pairs to the template. All DNA polymerases also require a primer to begin synthesis. They cannot begin without a strand of RNA or DNA base paired to the template. RNA polymerases do not have this requirement. They usually synthesize the primers. To build up a more detailed picture of replication, we first concentrate on the prokaryotic replication using E. coli as a model system. We then look at the eukaryotic replication, primarily in terms of deviation from the prokaryotic replication. There are a number of reasons why E. coli has been such an effective model to study replication. It has a small genome, which is 4.6 million base pairs of DNA. Its large collection of mutants that affect replication, and the circular chromosome is replicated at up to 1,000 nucleotides per second. In E. coli, replication begins at a specific origin called ORI-C, when a protein called DNA-A recognizes and binds to ORI-C. This causes an AT-rich sequence to be melted, making single strands available to load the enzymatic machinery for replication. An AT-rich region is easier to melt, as AT base pairs have only 2 hydrogen bonds compared to the 3 NGC pairs. So this shows an overview of prokaryotic replication. This is figure 14.13. Replication is bidirectional from a unique origin, and replication initiates from that unique origin. So 2 separate replisomes are loaded onto the origin and initiate the synthesis in the opposite directions on the chromosome. So to put it briefly, E. coli has at least 3 DNA polymerases: DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. In addition to adding nucleotides to a growing DNA strand, some polymerases also remove nucleotides or act as a nuclease. Enzymes that act as nucleases are classified as either endonucleases, which cut DNA internally, or exonucleases, which remove nucleotides from the DNA end from the end of DNA. So an enzyme with DNA unwinding activity is called helicase. And the unwinding of the two strands introduces torsional strain in the DNA molecule. So imagine two rubber bands twisted together. If you now unwind the rubber bands, what happens? The rubber bands already twisted about each other will further coil in space. When this happens, the DNA molecule is supercoiling. And from here, we'll move on over to section 14.5 with eukaryotic replication. Eukaryotic replication is complicated by two main factors: the larger amount of DNA organized into multiple chromosomes and the linear structure of chromosomes. This process requires new enzymatic activities only for dealing with the ends of chromosomes. Otherwise, the basic enzymology is the same. The sheer amount of DNA and how it is packaged constitutes a problem for eukaryotes. Eukaryotes usually have multiple chromosomes that are each larger than the E. coli chromosome. If each chromosome had a single origin, the time necessary for replication would be prohibitive. Eukaryotic cells reduce the time needed for replication by using multiple origins for each chromosome, resulting in multiple replicons. The origins are not sequence specific as in RIIC, and the recognition seems to depend on chromatin structure as well as on sequence. The number of origins used can be also adjusted during the course of development, so that early on, when cell divisions need to be rapid, more origins are activated. Each origin must be used only once per cell cycle. Prior to S phase, helicase complexes are loaded onto possible replication origins but not activated. Then during S phase, a subset of these are activated and the rest of the replosome assembled. The action of the replosome differs from that in E. coli in a few ways highlighted here. Priming is accomplished by a complex of DNA polymerase alpha and primase. This complex synthesizes primers consisting of RNA and a short stretch of DNA. There are also different polymerases for the leading and lagging strands. DNA polymerase epsilon is responsible for the leading strand, and DNA polymerase delta does the lagging strand. The sliding clamp subunit that allows the enzyme complex to stay attached to the template is called pCNA, for proliferating cell nuclear antigen. The name comes from its identification as an antibody-inducing protein in proliferating or dividing cells. The proliferating cell nuclear antigen sliding clamp forms a trimer, but has similar structure and function to the beta subunit sliding clamp. Synthesis on the lagging strand differs most from related events in prokaryotic DNA replication. The Okazaki fragments are significantly shorter, 200 base pairs instead of 1,000 base pairs. As each fragment reaches the previous segment, synthesis continues to displace the strand containing the previous primer. The displaced primer is removed by a nuclease, and the remaining fragment joined by ligase. The replosome, which can replicate both strands of DNA simultaneously, requires a variety of functions, including DNA unwinding, which is helicase, synthesis by way of primase and polymerase, and processivity factors, sliding clamps, and their loaders. There is clear functional conservation of these activities across all domains of life, but major components appear to have evolved independently in bacteria and in archaea eukarya. Members of the DNA polymerase family of enzymes have similar structure and homologous synthetic domains. They are differentiated based on non-homologous regions into subfamilies. The subfamily used by eukaryotes and archaea is the same, but different from the one used by bacteria. Replicative helicases all have the same structure, all have a similar structure, forming a ring around a single strand of DNA, but bacterial and archaeal eukaryote enzymes are not homologous and even move in the opposite direction along DNA. Lastly, the primase component of the replosome is not homologous between bacteria and archaea eukarya, not homologous. The function that is clearly homologous in all domains of life is the plasticity factors, sliding clamps, and clamp loaders. The specialized structures found on the ends of eukaryotic chromosomes are called telomeres. These structures protect the ends of the chromosomes from nucleases and maintain the integrity of linear chromosomes. These telomeres are composed of specific DNA sequences, but they are not made by the replication complex. Replicating ends are next. The very structure of the linear chromosome causes a cell problems in replicating the ends. The directionality of polymerases, combined with their requirement for a primer, creates this problem. Consider a simple linear molecule like that you see here in figure 14.21. Replication of one end of the template strand is simple, namely the 5' end of the leading strand template. When the polymerase reaches this end, synthesizing that 5' to 3' direction, it eventually runs out of template and is finished. But on the other strand's end, the 3' end of the lagging strand, removal of the last primer on this leaves a gap. This gap cannot be primed, meaning that the polymerase complex cannot finish this end properly. This result would be a gradual shortening of chromosomes with each round of cell division. When the sequence of telomeres was determined, they were found to be composed of short, repeated sequences of DNA. This repeating nature is easily explained by their synthesis. They are made of the enzyme telomerase, which uses an internal RNA as a template and not DNA itself. The use of the internal RNA template allows short stretches of DNA to be synthesized, composed of repeating nucleotide sequences complementary to the RNA of the enzyme. The other strand of these repeated units is synthesized by the usual action of replication machinery, copying the strand made by telomerase. A gradual shortening of the ends of chromosome occurs in the absence of telomerase activity. During embryonic and childhood development in humans, telomerase activity is high, but it is low in most somatic cells of the adult. The exceptions are cells that must divide as part of their function, such as lymphocytes. The activity of telomerase in somatic cells is kept low by preventing the expression of the gene encoding this enzyme. To examine the role of telomerase, mice were produced that completely lacked telomerase activity. These mice appeared to be normal for up to 6 generations, but they showed steadily decreasing telomere length that eventually led to non-viable offspring. This implies a relationship between cell senescence, or cell aging, and telomere length. Normal cells undergo only a specified number of divisions when grown in culture. This limit is at least partially based on telomere length. Support for the relationship between senescence and telomere length comes from experiments in which telomerase was introduced into fibroblasts in culture. These cells had their lifespan increase relative to the controls that had no added telomerase. Interestingly, these cells did not show the hallmarks of malignant cells, indicating that the activation of telomerase alone does not make cells malignant. A relationship has been found, however, between telomerase and cancer. Cancer cells do continue to divide indefinitely, and this would not be possible if their chromosomes were being continually shortened. Cancer cells generally show activation of telomerase, which allows them to maintain telomere length. But this is clearly only one aspect of conditions that allow them to escape normal growth controls. Despite the high fidelity of DNA, replication errors still occur. With no way to correct errors, cells would accumulate an unacceptable level of deleterious or lethal mutations. A balance must exist between new variation arising by mutation and the effects of deleterious mutations on the individual. In addition to errors in DNA replication, cells are constantly exposed to DNA-damaging agents such as radiation, UV light, and X-rays, and chemicals in the environment. Note to you all, please protect the skin you're in. Cover your skin, wear sleeves, wear pants, wear sunscreen. Wear caps, wear sunglasses. And please watch out for radiation from X-rays and chemicals in the environment. So those are agents. These are agents that damage DNA, and they can lead to mutations. So I say again, agents that can damage DNA can lead to mutations, and any agent that increases the number of mutations above background levels is considered a mutagen. Sunlight itself includes UV radiation and is thus mutagenic. Ozone normally screens out much of the UV radiation in sunlight, but some remains. Regions in the southern hemisphere show increases in skin cancer that correlate with a seasonal ozone hole, indicating a relationship between sunlight and mutations. We are also exposed to mutagens in our diet. I'll say it one more time. We are also exposed to mutagens in our diet as either contaminant or natural plant products that are mutagenic. When a simple test to detect mutagens was devised, screening of possible sources indicated an amazing diversity of mutagens in the environment and in natural sources. As a result, consumer products are now screened to reduce the load of mutagens we are exposed to, but we cannot escape natural sources. The process that can restore damaged DNA to its native state is called DNA repair. One indication of the importance of DNA repair is the number of repair systems that have been characterized. All cells that have been examined show multiple pathways for repairing damaged DNA and for reversing errors that occur during replication. These systems help to reduce the mutational load on organisms to an acceptable level. The most common forms of repair appear to have evolved early in the history of cellular life, indicating their significance. DNA repair falls into two general categories: specific and non-specific. Specific repair systems target a single kind of lesion in DNA and repair only that damage. Non-specific forms of repair use a single mechanism to repair multiple kinds of lesions in DNA. So this has been your instructor.